to bring up my special guests. So if I could bring up Jennifer Adams, Doug, and Michael McGuinness, that would be great. So we're going to play this one a little bit by ear. We have some questions which have been submitted, but equally a little bit conscious that you've only just finished listening to the last one. So we may end up with a few more as they come through, and we'll see where we go. So the first question I have is to start with Michael. So Michael, how do you decide what studies to perform inspections on?
That's trade secrets. I mean, it's fair to say it's a logical process, but it depends on the study type. So we have in the UK routine and triggered inspections. For routine inspections, we will look at the list of studies that have been submitted to us, evaluate where they are of interest. If you've previously been inspected to us, there might be specific aspects of those study conduct which we find would best show your compliance and a suitable candidate. Triggered is very different, obviously, because it's been raised as part of an application process and it's very specific and targeted. So yeah, it's not one factor. So it's not like a case of the fourth one down on the list or anything like that.
So yes, it's a double secret, trade secret. But I mean, like MHRA, it's a multitude of factors that you consider, including, the study, past history, or, if we have additional information, or there's certain things about the application that we want to take a look at or not. So there's a variety of different things that comes into play as part of that calculus. But yeah, it's double secret.
So then following on from that a little bit, so Doug, can you talk a little bit about how, or does the FDA complete BIOMO inspections for all NDAs and ANDAs?
Yeah, so we do not complete all inspections of NDAs and ANDAs. As part of the risk-based assessment, we try to do the best that we can, and we use it to site select, as mentioned before, using all the factors to go into whether it's an NDA or an ANDA. We're using a variety of factors to determine whether we go or not. Do we inspect in India? Yes, we do. A lot of our inspections are both NDAs and ANDAs. And there are a variety of different actors that we take a look at as well there too. So.
Thank you. Jenna, did you have anything to add on that one?
Sure, yeah. So the follow-up question to that was, do we conduct inspections from sponsors located in India? So we would inspect facilities regardless of the location of the sponsor if it was deemed necessary. That said, the bulk of our bioequivalence work is located in India. It's about a third of Office of Bioresearch Monitoring's total foreign work plan is spent within India. Specifically doing clinical bioequivalence inspections. I think it's fair to say that the analytical inspections, a lot of them are located in India probably as well.
Thank you. You can leave your mic on because I'm going to give the next one to you as well. So what's the role of paper source worksheets when the eligibility and the medical records and research activities are available in the subject's electronic medical records? For the person who asked the question, for their studies, they have the subject's medical record, ECRFs, and paper source worksheet. So they're worried a little bit about duplication of data.
And I think I would agree that this seems like a lot of duplicate entry. And anytime you have a single data point that's being recorded over and over again, you increase the risk of error and having discrepant data, which then requires more effort to explain. I would just say that, if you wanted to eliminate the middleman, in this instance, the paper source worksheets, you just would want to make sure you take a careful look at your EMR and make sure that it has all of the information that you actually want to capture, that it's accurate, that it's complete, and that it has adequate controls on the system. The last thing you would want is for somebody totally uninvolved in your study to be able to delete records that you are relying on for your source. So I would just make sure that you do your due diligence when assessing your EMR, but then I don't see any added value in transcribing that data yet. Again, you're only going to introduce more error.
Thank you. Michael, did you have anything to add on that one?
Nothing, just to echo obviously when you record something more than once, then it's definitely a risk factor. But yeah, I think for the sake of time.
Excellent, thank you. So next question, I'm going to hand it to Doug first of all. Do the inspectors look at the data generated, so things like pre and post study safety assessments by the clinical path labs when you're looking at BE studies?
The short answer is yes, we do. And we'll take a look at a lot of different things, and this is part of that kind of area that we can focus on and take a look at. But it's not going to be the end all be all. We take a lot. We look at that, but then we also weigh it against other things that we're looking at within the source documents or the study records as well. So.
And then Michael, do you want to chip into that one?
Yeah, I think Emma touched on this as part. The clinical laboratory values you usually get back can form part of subject eligibility, and you've seen the example that Emma discussed. So yes, we do review that. Emma mentioned that you do have both inbuilt pathology labs within the bioequivalence centres, but also external. So the level to which we review the data might be dependent on access, but certainly they form a key part of when we're doing that eligibility assessment. It's fundamental to subject safety to make sure you don't have anybody in the trial that's at possible risk.
Thank you. And I'll stick on the theme of clinical labs. This one is certainly something we've seen on inspection. So the question is, can clinical facilities have separate reference values for subject eligibility, which are different than the clinical laboratory reference, whether that's in an investigator's SOP or the facility's own list of values or protocol appendices? So perhaps you can kick that one off.
This one arrived late, so this is more off the cuff. The short answer is it will definitely depend. You can definitely see a scenario where a number of vendors are associated with a bioequivalence site, and the onboarding process will look at the difference between the laboratory reference ranges that those labs have and whether or not they make use of their in-house facilities, the in-house SOP-derived reference ranges. So one of the things that's crucial to consider on this point is inter-study, you're looking to compare apples with apples. So if you change a vendor during it, that is different to if you're using a single vendor for study A than when you might be using a different vendor for study B. We have, with bioequivalence centres, seen a variety of approaches to clinical reference ranges where the investigator's SOP overrides the accredited laboratory, usually expanding the acceptable ranges. And again, this is something that we'll query on inspection to look at what is that? Is that to, ultimate goal is that to extend recruitment and make it easier? But equally, is there any risk to subject safety? So it very much depends on the setup. And if the question was submitted in the room, I'm more than happy to talk in greater detail about the specific aspect, but it's a very good question.
I think the other key bit to that is that you almost have to stick with one. You can't pick and choose depending on what you feel like. So if you use one person's values to get somebody in and then use another set of values to keep somebody in potentially later on. So pick and stick with one. I couldn't help myself but give myself a question. I do apologise. Sorry, gang. So the question is, for the protection of human participants in a clinical trial or a BE study, is an insurance policy compulsory to come from the sponsor? So is it fine if there's no insurance policy, but the sponsor includes a statement in the master service agreement saying they'll cover any related injuries due to the study? In essence, that's a flat no. There's no self-insurance permitted. Doesn't matter who you are or what you have. That doesn't fly. You'll always need an insurance policy to cover your clinical trial. And you also need to make sure that there are no exclusions for each protocol that you run underneath it. Certainly, we've matched them up over the years and found trials which the insurance policy says you either can't be doing trials of this medication or in a particular group of participants. And lo and behold, that's exactly the kind of trials that we've found. The other thing is just to look for dates and that they actually cover the activities and the sites and things that you have listed. So insurance is always an interesting one, not particularly exciting to look through, but there's often plenty of things to find within it. The next question is for Doug. What's expected in terms of proof within source documents of not meeting the exclusion criteria? And in particular, studies which don't have chronic diseases, past medical records, those kind of things.
Yeah, so when most of the conversation that I had before was talking about inclusion, so where you have to have something before you can come in. When we're talking about exclusion criteria, it's a little bit different perspective. It's what things would take you out of the pool of becoming part of the trial. We would look at, the documentation as you screen, right? As what kind of questions are you looking at, or what kind of things are you talking to the subjects around, get a medical history, especially if it's like, let's say an easy one would be an exclusion because you're a diabetic, right? You would expect the CI or the clinical site to pull a history at least to kind of take a look, and then start going through the interview and doing the checkoffs to make sure that they are not a diabetic prior to enrollment. So the exclusion side is not as clear cut as having to show something, but it's also part process and part of your self-created documentation.
Thank you. Michael, did you want to add anything to that one?
No, it's just to say that it's very difficult to prove a negative. Within the clinical documentation, we do see, for example, text to say that check of exclusion hasn't been met. Particularly for bioequivalence centres, we do see it, but again, the level of documentation that goes in the background, it's something that we would check.
Thank you. The next one's to all of you, and it's sort of a link across all types of trials, really. So would you all agree that most observations in BE inspections are similar to investigational clinical trials? I'll give it to you, Jenna, seeing as you've got that puzzled look on your face.
I mean, the objective of the program is the same: human subject protection and data integrity. So I would say that, because those are the focuses, regardless of whether we're doing a new molecular entity study versus a bioequivalence study, we're going to find things in those same two buckets regardless. I don't know if I'll throw it to Doug.
So I would agree. I think those are the primary things, to ensuring the quality of the data that's generated at the site, versus, or on the same vein, is protecting those subjects that are enrolled in that site. How you get about that will primarily depend on the functions of the site, the design of the study, and the protocol itself. And those details are going to be kind of driven by that aspect of it. But the overall approach, depending, regardless of a BE study or any other type of study, it's going to be very similar.
Michael?
Yep, so fully agree with my FDA colleagues. It's interesting because we have the same objective. As we said, the inspector's aim is there to find anything that detracts from data validity, or anything where the welfare of the subject has been put at risk. It's fair to say, though, to expand that across the GXPs. I have the advantage of being in a multi-GXP team, and we share everything between us, the observations, people's, like we say, whether it's conducted in Glasgow or here in Maryland, people have similar traits, and it's taking those learnings and applying it across the board. I wouldn't restrict it to say the presentation topics we've heard today don't apply to you because you don't do generic applications, but you never stop learning.
I'll chip in a little bit as well. I think BE studies are a little bit different in some respects because they're so compact and they're so intrinsically linked. You have your participants, you dose them, you take some samples, you sit on those samples for a little bit while you finish the rest of them, and then you analyse them all. The trials in the wider world with the patient populations take years to get to that point, and they're huge generally. Whereas bioequivalence trials can be 40, 50, 60, 70 participants. So they're very different in terms of a speed kind of thing and that intrinsic link. And both parts are often done by the same organisation. So they do pose questions sometimes around access to data, the ability to look at things in different ways. Unlike some of the monster normal, in inverted commas, clinical trials that we see where there's thousands of vendors involved, multiple sites, multiple investigators, all these kind of things. So there are different things to think about as well. On top of, is the data good? Are the participants safe? Can we trust the data? So there are nuances to all of these things. Again, I'll throw the next one open to the group. Can you tell me a little bit about sort of collaboration for sharing observations across GXPs? And I'll widen the question and say about, is this being about collaboration between us as organisations? So can you talk to me, first of all, about collaboration within your organisation, and then secondly, about collaboration between us as a group?
I can kick us off and then happy for. So yeah, in terms of, if I've got this right, collaboration within our group. So as I said, we share everything that we talk about. We have team meetings. We try to ensure consistency between inspectors, so it doesn't matter whether you get myself, Jason, Emma turning up, that you get a similar approach. So collaboration through training, discussions, professional development is absolutely there. All of our reports are peer reviewed by a colleague that was independent of the inspection itself. So in terms of that aspect, it's something that we encourage. It's well supported by management. In terms of collaboration across the different agencies, if I've not skipped Jason's question, I think it's very true that you've heard almost for the last 3 days, the word collaboration has been the word of the workshop. We work very closely with our partners here in North America, both FDA and Health Canada, and it's fair to say that information is invaluable. Lessons learned, lessons shared. Again, we talked about the similarities of GCP inspections, the specific aspects of BE, and we hold quarterly meetings between ourselves. I'm very familiar with colleagues here at FDA, but also at Health Canada, and that information sharing is an excellent source. And then I think if I open it up at that point.
Yep.
So on the FDA side, we are not the same group. We have the disadvantage of being a separate program. FDA aligned our programs, I think, in 2019. So our pharma folks are actually in a separate program from our BIOMO folks. That said, when you conduct an inspection, part of your preparation for the inspection is looking at the inspectional history, and the database of all of our inspectional findings is shared across all programs. And so we have access on a BIOMO inspection if I'm inspecting a large bioequivalence facility that is also inspected under GMP. I have access to the full report, all of the observations, the names of the investigators who conduct it, and would have access to them and all of the data that they discovered on their GMP inspection to help inform my upcoming BIOMO inspection.
I mean, not much to add, but it's just in the case of FDA, the volume is kind of an ordeal, right? And so we continue to work collaborate with all the various offices, with ORA, within CDER, but it's just a huge amount of information and lots of inspections, lots of different types of inspections, but they do get pooled, and it is part of the calculus every time we site select, or when we go out to inspect, or when we do a review post-inspection. We're always looking at the totality of information. So that's a continuing thing, to continue the collaborations cross agencies. That's something we do all the time, and it's super fun. I think it's great, and it's a great way to share stories, share information, and like you mentioned, kind of learning moments about what you've seen here, certain types of inspections or different kind of things. So it's a valuable thing that I think has been going on for a bit of time, and it's something that I think everybody here is looking forward to continuing for the long term.
Thank you very much. I'm going to keep the next one because the answer is a bit of a negative, but the question was, is informed consent required for data taken from a biorepository? And we've taken that as sort of registry data where access to the patient may not be possible. So you don't know when the patient may next be coming in for a checkup, the patient's moved and didn't tell us, these kind of questions. So we've taken that as a BE kind of question, that you've had a volunteer for a while, and perhaps they haven't attended the last visit. And we've taken it as, could you then use registry data to potentially follow them up if they just didn't come back? The answer in some respects is we have no experience of that, of using registry data in bioequivalence trials, really. We'd want that clearly defined if ever you were going to do that, and it seems to be a fairly blunt instrument of, are they still alive, have they attended a hospital, and quite when you might be able to access that data, and even whether or not you have consent to do that. But just, I guess, be careful, but something to explore potentially in the future. I'm just going to give my poor panel one more question, and then I'm going to release you. The question's suitably broad, so by all means, feel free to go with it. The question is, can you please define oversight, study, and reporting? And so, Doug, I'll start with you.
Okay. So, oversight of study and reporting is, as you can imagine, huge, because it's, you're looking at different actors as part of the study. You're talking about the sponsor, you're talking about clinical sites, you're talking about clinical investigators, SSPs, CROs that are all in between. But what I would reiterate is that at every point or any touches within the study, you should have some overall oversight with what you're doing. And if you're the sponsor, you should have oversight in general of the entire study that's being performed. Those kind of controls should be built in a sense. It'll help prevent a lot of problems. It'll help, if there are problems, you've already pre-planned for them in how you approach them and deal with them. But the answer to this is, it kind of depends, right? Depends who you are and what you have to do. And then the reporting aspect of it will be related to that too. So it's a long-winded answer for me.
Michael?
Yeah, I can go next. So as Doug said, it's a huge, huge piece. It's in terms of a bioequivalence study, the oversight, one of the key pieces is, like we talked about, those touch points. It's not, we've got screening, we've got then getting them in, getting subjects or trial participants into the clinic. Linking them up with something like a lab where you might have a light-sensitive molecule, not having that two-way conversation, it's the same as pretty much any aspect of business. Communication is key. Oversight and control is a different aspect. Demonstrating it works differently in almost every company I've worked in or I've inspected. Some go with what I would call very militant, restrictive. We will meet every day at 9 o'clock. We'll have a toolbox meeting. We will go through this. Some don't. And there are strengths and weaknesses to both parts. The communication element is key, having information. The one thing is just to acknowledge the negative part of that. If you're a sponsor and you have outsourced everything to a BE centre, be aware of the role in which you as the sponsor are exerting your pressure and control. You looked during my bioanalytical presentation, if you'll allow me to jump into it, we saw the negative aspect of pressure on individuals where they pushed, and then we ended up with a data integrity breach, which then resulted in the trial ultimately going down. So what might start off as positive oversight, positive control, can very quickly be turned into a negative, and that's a very slippery slope to try and climb back up from.
Jenna, did you want to add anything?
I mean, I guess I will highlight that this is a great question. We don't spend a lot of time talking about oversight in bioequivalence compared to the first 2 days of this course. But ultimately, the oversight of the study at all of the levels that Doug has discussed, the oversight by the clinical investigator, oversight by the sponsor, oversight by the CRO, all of these different levels of oversight have a direct influence on the integrity of the data and the protection of the human subjects that come out of the study. And so as a result, it's just as important in bioequivalence as it is in a normal non-BE clinical trial.
And I'd like to link it back to Emma's presentation in some respects, picking up the reporting side of things. So it's important that the report bears resemblance to what actually happened on the trial. We've seen plenty of examples in the past where there are an errant cut and paste job with particular issues to this kind of thing, or you find things within the trial data itself which just don't appear in the clinical study report. And even when you're talking to people about it, they just don't necessarily appear. What's really important, and we talked about risk earlier on in the workshop, if things happen, if you tell us, we can think about it and give a considered opinion and try and link it up to how it all fits together and how it works. If you don't tell us and we find it, we've not really got too many options left to us. It might be possible to extend the inspection, but certainly if we're overseas, that's limited. Or the next one is to go back to the assessors and start to say, we have problems here. We found this stuff, it's not in the clinical study report. Over to you, what do you want to do with it? So be open, be honest, discuss, comment on what you found, talk about what you did when you found it and what you then put in place to try and deal with it. And then we can have an active conversation about what that is. That's really helpful to have it up front so we can prepare for it, talk to the assessors potentially. If it's in the CSR, If you've omitted to put it in an error and it comes up in inspection, then again, talk to us early. Let us know that there's something we need to work our way through. So I'm going to draw it to a close there. Thank you very much, panel. Thank you very much for coming to join me on stage. If I could ask you to put your hands together and thank the attendees. My final job is the important one, and that's to let you all go for lunch. We're back at 1 o'clock for lunch, so that gives you a bonus 12 minutes to grab a little bit of lunch. So thank you very much. We'll see you for pharmacovigilance this afternoon.